Welcome everyone to this webinar. It's great to see so many people joining us today. I'd like to start by respectfully acknowledging the traditional custodians of the lands on which we all meet today and pay my respects to elders past and present. I am joining you from the lands of the Wurundjeri Woiwurrung people of the Kulin Nation. Today, I will give an introduction, then we will have 4 presentations. We'll finish with a panel discussion at the end, so please put your questions in the Q&A box. This session will be recorded and shared. For those who don't know me, I'm Chris Elvidge, Head of Research at the Childhood Dementia Initiative. All of the work that the Childhood Dementia Initiative does is guided by families, including those on the screen now. All of these children have a childhood dementia condition. We have over 100 parents signed up to participate in our Family Advocates program to raise awareness and contribute to consultations and research. I want to acknowledge the parents who are joining us today, those currently caring, and those who are bereaved, and any extended family and friends. For some, the presentations today will be bittersweet because this research will be too late to help their children, but we hope that it is comforting to know that researchers both here in Australia and around the world are working towards solutions that will help children and families in the future. For those new to childhood dementia, I'll just give a brief overview. Childhood dementia is caused by over 100 genetic conditions that cause progressive cognitive decline. Collectively, they affect 1 in 2,900 births, and like dementia in adults, childhood dementia is progressive. Children lose the ability to walk, talk, read, write, and play. Most children have symptoms by the age of 2, and half will die by the age of 10. 70% die before adulthood. It's estimated that there are 91 deaths each year in Australia due to childhood dementia. And for comparison, 92 0 to 14-year-olds die from childhood cancer each year. You may have seen the webinar we hosted last year about the inequity in clinical trials and research attention. The take-home message was that the really devastating death toll from childhood dementia is because there aren't approved treatments or enough clinical trials. More research is needed. We've been advocating for more research funding since we started in 2020, and at our inaugural research symposium in 2022, the Health Minister, who was Greg Hunt at the time, announced an MRFF funding round for childhood dementia. At the next symposium in 2023, the Health Minister, who was Mark Butler, announced the recipients of that funding, 5 projects. Today, we'll hear about the progress of 4 of those projects. Unfortunately, Professor Wendy Gold couldn't make it today, but we look forward to hearing about her research another time. Acknowledging the varied audience that we have here today, all with different expertise, I'm going to give a little introduction that will show how the presentations fit together. All of the talks today are about different approaches to tackling childhood dementia. As I already mentioned, childhood dementia is caused by more than 100 genetic conditions. So we're going to start there. Childhood dementia conditions are monogenic, which means that a change to a single gene causes the progressive brain damage. There are more than 400 genes known to cause childhood dementia. So first you will hear from Dr. Nick Smith and Dr. Adeline Lau, who will talk about a gene therapy approach that aims to introduce a healthy copy of the faulty gene into the nucleus of the cell. The next target is the mRNA. mRNA is a working copy of a gene which is exported out of the nucleus of the cell into the cytoplasm, where the protein-making machinery is. If the mRNA is faulty, either no protein is made or a protein is made that doesn't work properly. You'll hear from Dr. Yahai Hong, who is working on an approach which involves introducing healthy mRNA into the cytoplasm of cells so that functional protein can be made. Finally, having a faulty protein or missing a certain protein can have a range of downstream consequences for the cells of the brain and the other parts of the body. Associate Professor Tony Cook will talk about an approach that aims to deal with the downstream consequences of the faulty or absent protein. Our final talk will be by Professor Michelle Farah and Dr. Arlene De Silva. They're going to talk about biomarkers, which are substances that can be measured in the blood or other bodily fluids. I'm sure they will tell you more about why biomarkers are needed, but if we continue our story from the previous slides, if testing any of those therapeutic approaches in clinical trials, it can be difficult to quickly and accurately determine if a treatment is working. This makes clinical trials long, expensive, and burdensome, and contributes to trials failing. Biomarkers could help solve this. Now I'd like to introduce our first speakers. I'm going to stop sharing now so that they can share their slides and turn their cameras on. Dr. Nick Smith is a consultant neurologist and the department head of neurology and clinical neurophysiology at the Women's and Children's Health Network in South Australia. He also holds an academic appointment at the University of Adelaide and is heavily involved in both preclinical and clinical research. Dr. Adeline Lau is a senior research fellow in the Childhood Dementia Research Group at the Flinders Health and Medical Research Institute. For over 20 years, Adeline has been investigating novel therapeutic approaches for the treatment of childhood dementia, particularly Sanfilippo syndrome. Nick and Adeline are going to present together on their project titled Developing Nanoparticle-Mediated Gene Transfer for Childhood Dementia. Take it away.
Thanks, Chris. Thank you to the CDI for inviting Adeline and I to speak today. I hope you can hear us, and apologies. It looks a little bit like we're giving an interview here from a very elongated room, but we hope you can hear us and see us on the screen. It's my pleasure today to be presenting with my colleague Adeline. For two reasons: she will speak far more eloquently than I will, and she will do her level best to keep me to task and on time for my tendency to ramble, for those of you that know me. So let's see how we go with that. We're very pleased to be able to speak today about the project for developing nanoparticle-mediated gene transfer therapy for childhood dementia. And in particular, we wanted to discuss this project in the context of one of the more common forms of childhood dementia, Sanfilippo syndrome, or mucopolysaccharidosis type 3A, which is one of the more common forms of childhood dementia. This is one of the lysosomal storage disorders, a form of diseases that are one of the great representations amongst the childhood dementia spectrum. And Sanfilippo syndrome itself being one of the more common of those, about 1 in 70,000 live births, and Sanfilippo syndrome type A being the most common of the four types, resulting from a defect in the gene SGSH encoding the enzyme sulfoglucosamine sulfohydrolase. And this is an enzyme that's critical for the catabolism, the breakdown of large molecules, glycosaminoglycans, within the body, and in particular within the central nervous system, and breaking down heparan sulfate so that this can be broken down and those molecules recycled and reutilized within the cellular system of the brain and the central nervous system. But of course, when that process is dysfunctional, these molecules accumulate and result in dyshomeostasis and dysfunction within the cells and within the neurons and supporting cells, with multiple perturbation and disruption of other cascades of other processes, inflammation, cellular inflammation, and other disruptions to the homeostasis of the central nervous system, resulting in progressive cellular dysfunction and organ dysfunction, which translates to clinical symptoms of dementia. And unfortunately for children with this condition, the most common form presenting in childhood from around 2 to 3 years of age, where children start to manifest behavioural disruption and a loss of developmental progress through progression to dementia, loss of skills, loss of motor skills, and from really around 3 years of age, very significant loss of both cognitive and motor skills to unfortunately a resultant early and terminal death in late childhood and early adolescence. Unfortunately, for this group of conditions, there are currently no approved therapies. And this is the case, of course, for many childhood dementias. However, there are some very encouraging gains that we are making in the approach to these monogenic disorders. And one of those approaches is the advancement of technology for gene transfer therapy. And for those that don't know about gene transfer therapy, just to recap on that, this is a technology that introduces a working copy of the dysfunctional gene, in the case of Sanfilippo syndrome type 3A, the SGSH gene, through the use of a delivery system, for the majority of cases using a viral vector system that delivers a gene back into the central nervous system, a healthy copy of that gene that utilizes then the host machinery to express that gene and to produce a functional copy of the protein that then is able to do the job that the endogenous protein is unable to achieve, and to restore function to that nervous system and hopefully provide a curative impact to the patient with the disease. And this is an approach that is being used certainly in Sanfilippo syndrome, but in a range of childhood dementias. And we have been involved in this area for a long time in developing these technologies. And the primary approach that has been used and has increasing global translation into clinical practice, and encouragingly we're seeing this in Sanfilippo syndrome. And you can see here on the slide a number of clinical trials that are in process, in particular on the top of this slide, the Ultragenyx pharmaceutical trial, which is perhaps the most advanced of these trials in this particular disease, and is coming towards, we hope, commercial delivery of this product for patients with Sanfilippo syndrome, is the use of a viral vector using an adeno-associated virus to deliver the transgene to patients with Sanfilippo syndrome. So this requires a virus delivery system, but there are some difficulties with this and some limitations with this, and this is the basis of the project that we're going to talk to you about today. But if we could cover why there are some difficulties with this process in the first instance. First and foremost, this process requires a virus to deliver the transgene. And these viruses are ubiquitous within the population, so we've all been exposed to them. The virus that is most commonly used for many of these technologies is a virus called adeno-associated virus. It's a single-stranded parvoviridae DNA virus of the parvoviridae family. Most people...
Can I just interrupt? Are you able to change the display settings so it's full screen?
Yes, it looks full screen on our system.
So we can see the slide that's coming up and the notes.
Swap presenter view and slideshow.
Yeah, try that. That's it, perfect.
Apologies for that. So the viral vector that we tend to utilize for this technology, as I said, is a adeno-associated virus, a member of the parvoviridae family, which doesn't cause any significant known disease within humans or primates. It's a fairly innocuous virus, if you will, but it's a common virus, and it's one that we within the general population are typically exposed to within childhood and certainly by adolescence. And indeed, most studies would suggest, globally, that up to 60% of the population have been exposed to this virus by late adolescence, adulthood. Some work by Tony Rasco here in Australia would suggest that about 44% of the Australian population have been exposed to adeno-associated virus by early adulthood. And indeed, our involvement for patients with the Ultragenics trial would suggest that about 34 to 40% of patients who we've been screening for involvement in that trial had seropositivity to this virus. And so what does that mean for patients and for this therapy? Well, unfortunately, it means that patients who are seropositive to this virus, they can't have that therapy. And the reason they can't have that therapy is that they mount an immune response to it. And that can have several effects. First and foremost, it can cause an acute immune response, which can have negative consequences to the patient and put them at risk of that therapy. And that's probably a minor effect and one that we can mitigate through other means. So that's not the major concern we have. But the bigger concern is that our immune system can develop neutralizing antibodies to this therapy. So by exposure to this virus, that very exposure creates a circumstance where we mount an immune response, which basically neutralizes the impact of that virus and negates its effect within the host. So it effectively negates the benefit of that therapy to those patients. And unfortunately, therefore, those patients derive no benefit from the therapy. And this is a significant issue because the evidence we're seeing is that gene transfer is having a positive benefit for patients for this disease in particular. And for many diseases within the childhood dementia spectrum, and we would project for many other diseases that are yet to receive attention from this approach, but have great potential for benefit from this approach. So the technology is a really important one, but this is a very significant limitation and one that we want to overcome. There are a number of ways to overcome that. One way, of course, is to look at how we might modify the immune system to mitigate the impact of neutralizing antibodies to patients. And certainly there's a lot of work being done around that. But one of the other approaches that is being looked into is how can we develop vectors and delivery systems that reduce the impact of the immune response from the host, so that neutralizing antibodies are removed from the picture, or at least the impact of that is reduced, and so that we can widen the number of patients that will benefit from these technologies. And so our approach to that has been to look at non-viral vector-mediated gene therapy. And so what do we mean by this? Well, effectively what we're saying is if we don't use viruses to package our gene delivery systems, our transgene, the product, the therapy that we want to deliver to patients, how can we package these in another way to achieve the same end without having to use an immunogenic virus? And one of the strategies that we're looking at to use for that is nanoparticle delivered therapies. So to use, in this case, a lipid nanoparticle vector, which is a lipid-derived coating. And I'm not going to go into the details of the specifics of that for the talk today, but a coating around our DNA delivery vector, our expression vector, DNA expression vector, that facilitates delivery of that vector to the target organ, in our case, the brain, that can be produced in the same and delivered in a similar way to the viral particles to achieve that delivery and that therapeutic effect. And so the primary advantage of this is that we can mitigate the immunogenic response, or at least greatly reduce that. But there are additional benefits too, in that this is often a scalable approach. It's a cheaper approach. and can lead to improvements in manufacture of these technologies. And indeed, this technology can be expanded to a range of other monogenic disorders. So we've developed a lipid nanoparticle, and we're in the process of doing that to test this approach. I don't want to go into detail here, but just to give you some details about the particle itself. These are small particles. As I said, they're a lipid-based product. They are what we call polydispersed in terms of size. They have a distribution of size that ranges between about 40 to 200 odd nanometers, with an average of about 100 nanometers in size. With an encapsulation efficiency, this means how much of the particles have the transgene inside them, the DNA cassette inside them, and it's about 90% on average. And we're working on ways to improve this efficiency. These are stable particles, and they're what we call polydispersed, so they have a uniformity of size and the characteristics that are what we're trying to achieve to deliver these products. And we have introduced these particles to a mouse model to demonstrate their efficacy for delivery of our gene to the brain of, in this case, our mouse model. So we want to show you some of this data. This is an experiment where we used our developed nanoparticle containing a DNA reporter gene, a GFP, so a fluorescent reporter gene, inside of our plasmid delivery vector within our nanoparticle. And we've delivered 3 treatments here: a PBS vehicle treatment, so just essentially the dilutant, the DNA plasmid itself, so the gene with the reporter gene without any nanoparticle, and then the reporter gene inside the nanoparticle. And we delivered this to the brain of a mouse through stereotactic injections to 2 regions at a dose, and you don't have to worry about this dose other than to know that it's roughly equivalent to the dose that we utilize for gene delivery in human gene delivery, for human gene transfer to mice to see whether we had any benefit or any delivery of that gene. So this is the vehicle-only delivery to the mouse brain. And for those of you who are unfamiliar with this sort of image, this is an image of a mouse brain cut in half, looking from the front of the brain to the back of the brain. And the little blue dots are the nuclei of the cells within the brain. And what we're looking for here is green. The gene that we've delivered to the brain should glow green. And on this slide, there is no DNA in here, so we don't expect to see any green rather on this slide. You can see a little bit of hazy green around the edge. You just have to take our word for it that is an artifact of the staining process. But if you can see on the magnified views, there is no appreciable green on this slide, as we would expect to be the case. This is a slide where we gave the DNA itself. So this does have the gene intact, present rather. And you can see that on this slide, there's a little bit of green uptake there. So there is a little bit of gene expression, but not very much. But this is the slide where we have the gene expression cassette packaged, or plasmid rather, packaged within the developed nanoparticle. And I trust that even for the uninitiated, you can see that there is significantly greater green on this slide. And in particular, you can see that at the back of the brain, this is a region called the cerebellum, where there's uptake in the Purkinje cells. And indeed, at the front of the brain, in an area, you can see the tract where the injection was injected, and indeed regions of the cortex where there is uptake within the neurons, that there is expression of that reporter gene. So very encouragingly, we have uptake of our nanoparticle delivered gene product here, and expression of that gene within these regions. So the particle has achieved what we were hoping to do, which is delivery of our transgene across the neuraxis of the brain, or at least at an appreciable level. So this gives us great encouragement that the nanoparticles, at least in their current iteration, are working to some degree. So this then leads us to our next phase, which of our study and what we'd like to briefly talk about now, which is to extend this nanoparticle experiment to look at a therapeutic transgene using the SGSH gene. You recall this is the gene of deficit in Sanfilippo syndrome, and to look at how effective these particles are for Sanfilippo syndrome within the disease state and for correcting that disease. So I'll hand over to Adeline to take you through some of that work.
Thanks, Nick. So to begin with, we use the same vector backbone. It's a very small, minimally sized vector, and we kept the same promoter that we used for the GFP studies, but cloned in the human sulfamidase gene, and then we wanted to confirm that that would increase the expression, so we transfected some 293 cells. And as you can hopefully see in the graph here, the untreated and mock-treated cells in the black and pink have very little expression, whereas our cells that were treated with our DNA containing the sulfamidase in purple have much higher levels of expression. Again, similar to before, we characterized their biophysical characteristics, and in the interest of time, I won't go through them completely, but I just want to say that they were very comparable to what we saw with the GFP product. So there's a lot of consistency between the batches, and we are working towards, this is a manual process at the moment, so we're working towards more automated methods to improve these characteristics. So using the same study design as we'd used in the GFP study, we delivered the PBS vehicle control, the naked DNA, or the sulfamidase DNA within the nanoparticles to mice. So we employed our naturally occurring model of MPS IIIA. We've had a lot of experience with this model. And again, we injected it into 2 brain regions on both sides of the brain, so 4 needle tracks in total. And we delivered these to the MPS IIIA mice at a preclinical age of 6 weeks of age, closely monitored them for 4 weeks, and then collected various tissues and fluids, including the brain, where we divided the brain in half. One half was taken through for histological staining, and the other half was divided into further slices and taken through to biochemical assays. So unfortunately, in our first test here, when we looked at biochemical assays, the graph on the left is the enzyme activity. So this is the enzyme that's deficient in MPS IIIA. You can see that the unaffected healthy mice have a higher level of enzyme compared to our affected MPS vehicle and DNA controls. And the purple nanoparticle treated mice didn't have any improvement on a whole brain slice level for activity. And so unsurprisingly, when we measured the amount of heparan sulfate disaccharide, so the storage product that this enzyme breaks down, we didn't see any impact on the amount of heparan sulfate storage within these brains. We did think, however, though, the way we section these mice, this was that entire brain slice. So potentially there would have been regions of improvement that were very close to the injection site that might have been diluted out by other cells within that same slice that might not have had a treatment effect. So what we did was immunostained for lysosomal interval membrane protein 2, or LIMP2. This is another lysosomal marker. And as you can see, hopefully in the graphs here, you've got the unaffected healthy mice in black. These are the three different graphs indicate 3 different brain regions that are close to the injection sites. So we have low levels of this lysosomal marker in the black in the healthy mice, higher levels in the MPS vehicle mice in the pink, and there was a trend towards a slight reduction in our nanoparticle treated mice in the purple, but this was a non-significant trend. Keeping in mind this data was taken from mice that had only been treated for 4 weeks, we considered whether or not improving the amount of time, so that increasing the length of time that these mice were treated might have a better therapeutic effect. So we took an additional cohort of mice again delivered the nanoparticles to our MPS IIIA mice, but instead of taking samples at 4 weeks, we extended this time point out to 15 weeks instead, and again divided the same dissection strategy for the brain for biochemistry and histology. Of the samples we analysed, our nanoparticle treatment didn't have any effect on the body weight gain. So our affected mice have enlarged livers and spleens as part of their disease phenotype. And our therapy didn't have any impact on that increased body weight. We also didn't see any difference in the biochemical markers, so the brain sulfamidase activity or the heparin sulfate, which are the two panels on the top there. And we also didn't see any reduction in that LIMP2 immunohistochemical staining. So they're the two different brain regions in the bottom two panels. Though again, we saw a slight decrease, non-significant trend in the purple versus the pink. So we considered whether or not our particles might have had some sort of immune response. So we've begun screening we had sera samples from these same animals. So we've screened them against various lipids that are part of our particle composition for IgM antibodies, and we've not seen any antibodies detected so far in our younger mice, and we will follow this up with antibody testing against the sulfamidase product or lipids for IgG antibodies in the other cohorts. And so just to summarize the data presented so far, this novel formulation, it generates very consistent, small, stable, and uniform sized nanoparticles. We can show that there's increased activity of our product in vitro and cultured cell models. With our GFP, we have beautiful widespread transgene marker expression throughout the neuraxis delivered to the healthy mouse brain. However, when we do the same treatment strategy in our MPS IIIA mice with our sulfamidase nanoparticles, we didn't see impact on our biochemical markers or histological markers for 15 weeks post-injection, though there was the non-significant trend in regional reduction at areas close to the injection site using immunohistochemical staining. So where does that take us now? We're still actively working on this project. So what we do know is that our MPS IIIA mice have a very strong and robust neuroinflammation. It's very early in the disease course. And so our current thinking is potentially this this highly immune reactive environment has impacted the transgene expression in our MPS IIIA mice. So the GFP studies were delivered to healthy mice. So potentially if we immunosuppress affected mice at the time of treatment, we may be able to have improved transgene expression there. We're also generating new plasmid constructs with a bioluminescent reporter, so we can actually monitor the transgene expression in lifetime. So we can repeatedly anesthetize the mice and see where the expression is and the kinetics of expression and distribution of expression. And we're trying to improve our particle production methods as well. And so just finally, I've tried to knowledge everyone as we've gone on the slides. This has been a very large collaborative project. There's been lots of students and staff involved, lots of course facilities. So we'd like to thank everyone that's participated in the project, as well as the Childhood Dementia Initiative and St. Philippa Children's Foundation, who also supported the grant application, as well as the funding from the MRFF. Thank you.
Thank you, Nick and Adeline. That's great. Now, if you'd like to stop sharing, and I'll introduce our next speaker. Dr. Yahai Hung is a Senior Research Fellow at the Florey Institute in Melbourne. Today, she's going to talk about the development of mRNA gene therapy for Niemann-Pick disease type C. And sorry, I'm just clicking the button for it to allow her to share. And she's going to also talk about how this could be a blueprint for other childhood dementias as well. Yeah, hi, do you want to put your camera on and share your screen?
Yes, sorry, Chris. Give me 2 seconds. I just need to. Yes, thanks, Chris, for the introduction. And also thank you to Childhood Dementia Initiative for this opportunity to share an update on our project here today. So here is the project team. We are a multidisciplinary team, and we all have a common goal of improving the health of children living with dementia. And through this project, we are hoping to be able to build an mRNA gene therapy platform that we can then apply to all childhood dementia conditions with a monogenic mutation. So first of all, some background on the mRNA technology that I'm using to develop gene therapy for Niemann-Pick disease type C and also other childhood dementia conditions. So by now, everyone is familiar with the term mRNA. So in 2021, mRNA became a household name when FDA first approved the first mRNA medicine, the COVID-19 vaccines, showing that synthetic mRNA could be formulated, manufactured, and also delivered safely at a global scale. So this achievement not only saved millions of lives, but also brought to the world's attention mRNA's therapeutic potential. And 2 years later, in 2023, the Nobel Prize honored Professor Caroline Carrico and also Drew Weissman for their pioneering work on mRNA stabilization. Their discovery has not only enabled the mRNA COVID-19 vaccines, but it is now also used in other mRNA therapies and developments, including the gene therapy platform that we are developing here for childhood dementia. And the timeline here on the right shows that the success of COVID-19 mRNA vaccines did not happen overnight. So since the first discovery of mRNA in 1961, there has been 60 years of incremental advances in enzymatic synthesis, chemical modifications, and also delivery vehicles, such as the ones that Nick and Adeline are developing, have led to the robust mRNA technology that we know today. So Nick has already spoken about gene therapy, and traditionally that it is delivered using modified virus, such as adeno-associated virus or AAV virus, being the most popular to treat genetic disorders. But why mRNA, and why is it attractive? Why is it an attractive alternative to the AAV delivered gene therapy? First, it can be delivered cell-free, which means that this can be simplified in terms of manufacturing. Second, mRNA can be synthesized more rapidly compared to the AAV delivered gene therapy, which requires complex manufacturing processes. Importantly, mRNA does not integrate into the host genome like the AAV factors, so that there is no risk of insertion or mutagenesis that can cause off-target adverse effects. And also you can see in the table on the right that mRNA therapies can be far more affordable. A single dose of an mRNA COVID-19 vaccine currently costs $130 And compare this to the approved AAV delivered gene therapies like Luxturna and Zolgensma, this can cost up to $2 million for a dose. And what I find most attractive about mRNA is the ability to deliver personalized dosing. And what this means is that we can precisely control the amount of the therapeutic mRNA that is being delivered, tailoring the treatments to each patient's needs based on their biochemical and also genetic profiles. So how does the mRNA technology work? So Chris has already given some background on mRNA. So just to remind you again, the diagram here shows the flow of the genetic information going from DNA, which undergoes a process known as transcription, where the genetic information carried on DNA is transferred onto messenger RNA or mRNA that carries a working copy of the genetic information that it then undergoes a process known as translation to then produce the protein. So for mRNA gene therapy, we are looking at this pathway here in this flow of genetic information, where we can give cells instructions to produce healthy copies of protein using synthetic mRNA. And this would then let us fix a problem where there's a missing or faulty protein due to genetic mutation without changing the blueprint that exists on the DNA. And the cartoon that I show here is the protein, the MPC1 protein that is involved in the Niemann-Pick disease type C. And I'm focusing specifically on MPC1 protein at the moment, even though there are 2 genes involved in causing Niemann-Pick disease type C1. This is because MPC1 mutations causes 95% of the Niemann-Pick disease type C cases. MPC1 is a cholesterol transporter, so it is essential for cholesterol processing in cells. And using microscopy, we can visualize cholesterol inside the cells using a cholesterol binding compound known as vilipin. So on the left, if you look at the microscopy image down the bottom, we have healthy cells that has been stained with vilipin showing very little staining, indicating that there is low levels of cholesterol under healthy conditions. On the right, MPC patient cells, they do not have functional MPC1 protein, so they are not able to process the cholesterol inside the cells, and they have dramatically elevated levels of cholesterol. So to correct this, we are delivering mRNA that carries instructions for making a healthy copy of the MPC1 protein. So this slide is just to show you that we are able to make the DNA template that is then used to make the synthetic mRNA. And when we introduce the synthetic mRNA to MPC cells, which does not express any MPC1 protein, as you can see here in the Western blot on the right, it is able to restore the MPC1 protein production, demonstrating that our MPC1 mRNA therapy strategy works as intended. Furthermore, we are able to show that the MPC1 protein produced from the synthetic mRNA is able to normalize the cholesterol levels in the MPC patient cells, indicating that it is active and functional. So these results are really exciting and validate our approach of using mRNA-based gene therapy to treat Niemann-Pick disease type C and also other childhood dementia conditions that are monogenic disorders. However, to translate mRNA gene therapy to the clinic, especially for childhood dementia conditions, there are still a number of challenges that we must overcome first. So here are 2 key challenges that my team is currently working on. So one, mRNA and protein stability for sustained therapeutic effects, and 2, efficient delivery of the therapeutic mRNA to target tissues and especially to the brain. So first, looking at our challenge for mRNA and protein stability. So both synthetic mRNA and protein, the MPC1 protein, have a half-life less than 2 days. So what this means for the patients, if we are to deliver mRNA gene therapy, they will need regular injections every 2 days. And indeed, giving the MPC cells a single dose of mRNA, you can see here, after the initial spike in protein expression, we see a steady decline in the MPC1 protein expression over time. So mRNA therapy is not yet available as an oral dose, which means that patient will need to make regular visits to the hospital for intravenous injections to receive the mRNA gene therapy. So we want to reduce the dosing frequency so that the longer lasting mRNA means less visits to the hospital for the patients, less stress for the families, and also a more sustained therapeutic level of mRNA in the body. And the schematic diagram here is that of mRNA. It shows that there are 5 basic structural regions. In the middle, we have the coding region for MPC1 or any other protein of interest, and this flexibility to be able to swap out the gene of interest in the middle here makes mRNA an ideal platform technology for developing gene therapy for childhood dementia. And flanking this central region are 4 structural regions, and all of these structural regions are amenable or can be engineered to improve the mRNA stability and also the protein stability, so that one dose can last longer and reduce the dosing frequency that's required for the patients from days to weeks and hopefully perhaps months. So currently working on these projects are two students from my group, Jessica Zhang, who started her PhD today, and Roshan Dhillon, who is a second-year master's student. And we have a couple of lead mRNA candidates that are showing greatly enhanced stability that are currently undergoing further characterization. However, unfortunately, due to IP protection issues, we cannot share any details or data at this stage. But what I can tell you is that our data is quite robust and reproducible. And also looking at further improving the efficacy and the specificity of our mRNA gene therapy for childhood dementia, we have established a collaboration with Professor Wei Xiang and also Lillian Lagarig from the Australian Centre for AI in Medical Innovations to develop a generative AI model to optimize our mRNA design. And this is a newly established collaboration, so the work is in progress. But we are hoping that once we have validated our AI model, it will be then made publicly accessible to everyone to use. So we know that NPC is a childhood dementia, which means it's a neurological disorder. And the image here shows the key brain regions that are affected in NPC and the associated symptoms, which include cognitive decline, motor disabilities, ataxia, and also psychosis. So to restore the neurological function for NPC and other childhood dementia conditions, therapeutic mRNA needs to reach the brain. However, the brain is protected by the blood-brain barrier, and 99% of the drugs cannot pass through. And naked mRNA, due to the negative charge, cannot cross the blood-brain barrier. And it is also degraded quite quickly by the circulating nucleases. So these are enzymes that can break down mRNA that are unprotected. So here is our second challenge. How can we deliver therapeutic mRNA into the brain? So to address this problem, we're investigating two different delivery systems. One is using natural lipid nanoparticles known as extracellular vesicles. And our second approach is similar to what Nick and Adeline are doing, is to look at synthetic lipid nanoparticle formulations to deliver our mRNA. So both systems ideally should offer the following properties: protect the mRNA from nuclease digestion, also protect mRNA from unwanted immune interactions that then lead to inflammatory side effects. They should also have target specificity to minimize off-target effects and a broad distribution in the body, including ability to reach key brain regions. And also importantly, able to achieve what's known as endosomal escape to allow the release of the mRNA into the cell to make the desired protein, and in our case, MPC1 protein. So we are testing mRNA that are loaded into these delivery systems in a mouse model of MPC1. So a little bit of background on extracellular vesicles or EVs. So these are natural delivery system. They are small fat bubbles that are released by our cells. They are involved in cell-to-cell communication, shuttling proteins and genetic materials such as mRNA between cells. And they can be isolated from a number of different biological sources, including blood, urine, milk, and saliva. Therefore, they are highly biocompatible and can evade immune detection. And here is an electron microscope image of EVs, and the red arrows point to what we would typically expect to see of EVs. To load mRNA into EV efficiently to achieve a concentration that is suitable for therapeutic use has proven to be quite challenging. And we are currently working on overcoming this challenge. And if we are successful, EVs could be an elegant and biologically relevant delivery vehicle for MPC1 mRNA directly into cells where they are needed the most. So moving on to our second approach, which is using synthetic lipid nanoparticle formulations. So in contrast to EVs, lipid nanoparticles are approved for clinical use. So these are the formulation that has been used to deliver the COVID-19 mRNA vaccines. And typically, lipid nanoparticle formulations are made of 4 basic components. These are cholesterol, which are important for the lipid nanoparticle stability, and also structural integrity, lipids, which can change its charge to a positive charge under low pH, such as that in the cells, and it helps binding of the negative charge mRNA tightly inside the lipid nanoparticles, and also helps the lipid nanoparticles to release the mRNA into the cells once they're in the low pH environment. The lipid nanoparticles will also include a helper lipid, and this prevents the lipid nanoparticles from clumping and also extend their circulation time in the blood so that they have a better chance of reaching target tissues. And the 4th components of the lipid nanoparticles are known as phospholipids, and this contributes to the stability of the lipid nanoparticles, and again also help them to survive longer in the bloodstream. And Mahi Matri is a 2nd-year PhD student from my team, and in collaboration with Professor Colin Pelton at Monash Institute of Pharmaceutical Sciences, we are working on developing novel lipid nanoparticle formulations that can deliver mRNA into the brain that can deliver lipid nanoparticle formulation that can deliver mRNA into the brain. And we have exciting data that show the feasibility of this approach to deliver MPC1 mRNA into the mouse model, and we are seeing MPC1 expression. However, again, I am not allowed to share any data until we have our IP protection in place. But what I can share with you is our next step plans. So currently we are here at the stage 1 of the project. We are characterizing the mRNA distribution and also protein expression in the mouse model to identify the most optimal delivery routes and also the optimal dose to give to the mice. The next stage will be to determine the dosing frequency, which will then be used to inform our 3rd stage of the study, which is to evaluate the therapeutic efficacy. And for this, we will be assessing for improvements in the lifespan, improvements in cognitive and motor functions, and also biochemical changes in the blood and tissues collected from the mice. So in summary, I've shown you that in patient cells, using synthetic MPC1 mRNA, we are able to restore protein expression and also normalize the cholesterol levels in these cells. And we are currently improving our therapeutic mRNA's design to enhance the stability of mRNA to minimize the dosing frequency that the patient would need. And also we are working on optimizing our lipid nanoparticle formulation for delivery of the mRNA to the brain. And we're investigating 2 different delivery systems, natural lipid nanoparticle system known as EVs, and also synthetic lipid nanoparticle formulations. And we're currently testing our mRNA gene therapy candidates in the MPC1 mouse model. So what's for the future? So following MPC, we are now in the process of starting to look at mRNA gene therapy for another childhood dementia condition, Menkes disease, which is a severe copper deficiency disorder that affects mainly boys. And this will leverage my expertise in Menkes disease biology from my PhD studies. And ultimately, our goal is to build an mRNA gene therapy platform for childhood dementia and realize life-changing mRNA gene therapy to give children with dementia a chance to grow up healthy and live their dreams. And finally, I would like to acknowledge all the collaborators on this slide, and there are many of them, so I'm not going to go through each one of them. But most importantly, I would like to thank the Childhood Dementia Initiative for their advocacy for the MRFF funding, and also to the NHMRC for the MRFF funding, and also to the amazing Australian NPC Disease Foundation and the NPC patients and families in Australia for their support of this project from day one before COVID-19, before most people have heard of mRNA. And before I finish, I have two quick announcements. So if you would like to learn more about NPC, please come and join me at the 2025 Australian Limipic Conference next month. That will be held here at the Florey in Melbourne. Registration is open now on the Australian NPC Disease Foundation website. And I'm also really excited to share with you our new Ideas Lab project with ArtPlay, which is called Amazing Brain. And this is in collaboration with a visual artist, Luisa Buffadecci from the Victorian College of Arts. And this will be an immersive and interactive brain maze. And we are hoping to use this opportunity to raise childhood dementia awareness and also to provide an opportunity for children aged between 7 and 13 who are living with childhood dementia to share their lived experience through arts. So we will be looking to recruit children to help us to create the Amazing Brain art installation. So more details will be coming soon, and please let me know if you're interested. And here are my contact details. And thank you all for your attention.
Thanks, Yahai. Okay, I will now move on to our next speaker, Associate Professor Tony Cook. Feel free to share your slides and turn your camera on. He's head of the Stem Cell Models Group at the Wicking Dementia Research and Education Centre at the University of Tasmania. His research focuses on both childhood dementia disorders such as Batten disease and adult onset conditions as well, such as motor neuron disease. He's going to speak today about a new substrate reduction strategy to treat multiple childhood dementias. Thanks, Tony.
Thanks very much, Chris, and thank you to CDI for the opportunity to present the work today. So, why do we need a new substrate reduction therapy for lysosomal storage disorders? So we're really building off the promise of a drug called miglustat that some of you may have heard of. This is a drug that is used for some childhood dementia diseases, including Niemann-Pick type C that Yahoi just spoke about, and is currently in clinical trial for CLN3 disease, which is a form of Batten disease. But this drug has a fairly poor blood-brain barrier permeability. Only about 10% of it that's taken via an oral dose actually enters the brain. And there's a number of quite common side effects. So you can see them listed there. So for example, diarrhea occurs in about 85% of people taking this drug. And that's attributed to miglustat inhibiting the enzymes in the intestine that digest sugars, and so that leads to the diarrhea. And these side effects do contribute to people discontinuing treatment with miglustat. And so even though we know that it has some potential in stabilizing the disease, these side effects are detrimental to them continuing with treatment. There are other drugs that are similar to miglustat, but they have even lower brain permeability, and they're not currently in clinical trials. So there's this unmet need to kind of take the learnings that we know about miglustat and translate it into a more effective therapy. And so what we've done is to think about other ways that we can target the same pathway. And so on this slide here, down the left, you can see what we call the flow of genetic information that we've heard about in the previous talks. But down at the bottom, we have a generic drug inhibiting the activity of a particular protein. And so that is what most of the drugs that are available to us do. They act to inhibit the activity of a protein, and therefore that activity is not present. What RNA drugs do is to, instead of targeting the protein, they target the RNA in various ways to alter gene expression. And so the end result of a small RNA drug is to decrease the amount of protein that's present. And so that gives us the same functional effect as a drug like miglustat, where we're inhibiting the protein activity, but we've got no protein or drastically reduced levels of protein, and therefore a similarly reduced protein activity. So what are the advantages of these sorts of small RNA drugs? Well, they can be much more specific than small molecule inhibitors. They're able to produce long-lasting effects from a single dose. So typically after a few loading doses of these sorts of drugs, dosing can be anywhere from monthly through to sort of 3 monthly intervals. They do have, for neurological diseases, there's an alternative delivery route, and this is important when we consider the limitations of miglustat in causing diarrhea by inhibition of intestinal enzymes. Small RNA drugs tend to be delivered by intrathecal injection, and therefore they bypass the GI tract, and therefore are likely to overcome that limitation. And there's several of these sorts of drugs already in use, some for spinal muscular atrophy, a recently approved one for SOD1 associated ALS. And these seem to be well tolerated, even in children who receive these drugs. The adverse events are typically related to the injection itself, or are considered part of the normal course of the disease. So they're not associated with the actual small RNA therapy itself. So what we set out to do is to build on what we knew about miglustat and the biochemistry underlying its action, and see if we can use this new sort of technology to overcome the limitations of that drug. And so, as with all the speakers today, we were fortunate to receive funding from the Medical Research Future Fund. We have a broad range of expertise on the team, either as investigators or associated investigators, and we had fantastic support from 5 patient organisations, including CDI, but also for several other diseases where we think that this approach might be worthwhile as well. So let's have a look now at how we think this might work. And so if we can start on this slide down at the bottom, in the middle, you can see in white ceramide. And you can see coming out from ceramide are lots of these purple arrows, and these are indicating biochemical reactions where ceramide is metabolized, and it's built into gradually more and higher order complex lipids. And you can see up here there's several ones called GM1 ganglioside, GM2 and GM3. And they're the ones, at least for this talk, that we're interested in. And what we know about these gangliosides is that they are very important for proper neuronal function, but they accumulate in many lysosomal storage disorders. So next to all of the purple arrows, you can see orange arrows pointing in the opposite direction. And next to those in orange text is the name of various lysosomal storage diseases, many of which cause childhood dementia. And what these particular diseases are caused by mutations in the enzymes that break down these fat molecules. So, for example, in GM1 gangliosidosis, the enzyme that degrades GM1 ganglioside to GM2 ganglioside is inactive, and so you get an accumulation of the GM1 ganglioside. So we anticipate that if we're successful in this project, that the therapy will be useful for a range of diseases. And I've grouped them here down the left-hand side of this screen based on them either being important for, well, rather being involved directly with genetic or biochemical evidence that links them directly to ganglioside metabolism. Or there's a range of diseases where we know from preclinical studies or indeed clinical use of miglustat that this approach might be useful. And then the bottom grouping there are diseases that are sort of in and around this same sort of metabolic pathway and might also benefit. So miglustat works here by really inhibiting this first step in the synthesis of these more complex ganglioside fat molecules. And so it inhibits this enzyme called UGCG. And I'll do my best to not stumble over that protein name for the rest of the talk. So we hypothesized that, well, miglustat has some promise in inhibiting UGCG. Why don't we try and leverage this emerging small RNA technology and see if we can come up with a way to get around these side effects of miglustat that cause people to stop taking the medication. So we set out to ask 3 research questions. Firstly, can we design an ASO that decreases UGCG expression? If we can, does it also then do what we expect it to do, and that's decrease ganglioside levels? Ideally, does it rescue cell functions that we know are associated with disease? And can we do it in a way such that it does not adversely affect cells? And then what we want to do is then compare that or benchmark our new approach to the effect of miglustat. So what we've started with is a disease model of Tay-Sachs disease. And so this particular model that involves the conversion of GM2 ganglioside to GM3 ganglioside. And what we have on the left here are some fluorescence microscopy images where the cell nucleus is shown in blue and the GM1 ganglioside is shown in orange. And what I hope you can appreciate is in this top panel here, these are neurons with the gene variant that causes Tay-Sachs disease, and that there's a fair bit of orange staining in this image. But when we look at isogenic neurons, so these have been generated by gene editing, so they're genetically similar to the Tay-Sachs neurons, but do not have the gene mutation. I hope you can appreciate that there's less of an orange stain in that particular image. And so what we're looking for is an ASO that can kind of reproduce that sort of data. So the first thing we did was to test that our ASO can change expression of UGCG. And you can see in that middle panel there, we've tested several potential ASOs as well as several controls. And I hope you can appreciate that there's a couple in the middle there that show that the level of UGCG has decreased. We then went and delivered these ASOs into the Tay-Sachs neurons and stained the cells for GM1. And so this was done 4 weeks after a single delivery of the ASOs into the immature neurons. And so when we used a control ASO, the level of GM1 staining was quite similar. But then with our two most promising lead candidates at this point, we saw that there was a decrease in the intensity of GM1 staining in these cells. So this was quite exciting. We next asked if there were any functional consequences for Tay-Sachs neurons in this. And for this, we use a technique called electrophysiology, where we looked to quantify how the cells, how those neuron cells communicate with one another. So in our brain, we have nerve cells that can pass information in the form of electrical activity that we can then measure and quantify in various ways how the activity profile looks like. And so we've done this, again, using that same Tay-Sachs disease cell model and the same antisense oligonucleotides. And what we found in the blue was that the corrected, so these are the isogenic gene-corrected cells, compared to the orange, there were quite substantial differences, but with particularly with the second of our ASOs, we were able to start seeing some of these functional differences be rescued and returned back to a more normal cell phenotype. This didn't happen for every single parameter in these experiments, and so I'm just showing you some of them here. So by way of coming back to our research questions, can we design an ASO? Yes, we can. It seems to show some sort of promise at the moment, but we have more work to do to quantify some of the measures that we've been using and to check that there's no adverse things occurring in these cells. And then we need to do the sorts of head-to-head experiments using Milstat as a benchmark. And to do this work, we're moving forward with other stem cell-derived models. You can see them listed on the screen here. These are derived from various lysosomal storage disease patients with a range of genetic etiologies. And these are really powerful tools because we can mature them not just into neurons in the laboratory, but we can also start looking at other different cell types that are present in the brain and asking what happens to these cell types. But there's also a large number of diseases that we don't yet have stem cell models for, and so we can look at basic biochemistry using skin cells from a larger number of conditions more rapidly. And so that's where we'll be going as well. And so it just leaves me to thank everybody who's been involved in this particular project. I've highlighted on this slide Emma Wilkinson, who is a postdoc in my group here and has generated the data that I've presented using ASOs that have been designed by May Ong-Tut and her team over in Murdoch University in Perth. But again, this is a large collaboration between lab-based researchers, clinicians in pretty much every state of Australia, I think, are involved in this in some way. And we've had fantastic support from several childhood dementia-focused NGOs. And our work on the ASO has been funded by the MRFF, and some of the work on the different types of brain cells has been funded by Batten disease. So thank you very much, and please reach out if you have any questions.
Thanks, Tony. We're running a bit behind on time, so I'll move straight on to the next talk. Please put your questions in the box so we're ready for some quick questions at the end. We'll fit in as many in as we can. So now it gives me great pleasure to introduce Professor Michelle Farah. She's a child neurologist at Sydney Children's Hospital and a clinical academic at UNSW Sydney. And she leads a comprehensive, integrated and multidisciplinary research program. Also, she will be co-presenting with Dr. Arlene De Silva, who's a senior postdoctoral research fellow at SCHN and UNSW. Her research focuses on omic technologies to identify protein signatures related to disease activity. Over to you, Michelle.
Thanks, Chris, and thank you to Child Dementia for hosting today. And good afternoon, good morning to everyone, depending on where you are. So I think this is a nice follow-on hearing about the therapies that are promising. It's really important that we are able to have the best tools possible to be able to determine whether they're effective, early signals of that, so we know whether to continue or whether to stop, and also to evaluate safety. So our research is focused on looking for biomarkers to help with clinical trials and monitoring of treatments for childhood dementia.
Michelle, can I just interrupt? I can see your notes slide. Can you just change the display settings? And also, Tony, you might want to turn your camera off.
Yep, sure. I share screen too, that would be better. Is that better?
That's perfect. Thank you.
Thanks, Chris. I'm not very good at technology. So I think clinical trials are challenging because, you know, and I think they're challenging particularly in conditions that are rare and also in child dementia, because there are so many different individual conditions that fall under the umbrella. And because they're rare, there's small patient numbers. So the aim of a clinical trial is to have enough evidence to say this is a medicine that's effective, that should be marketed, and all children should be able to benefit from it. So to be able to do that, it needs to have, usually for a treatment to be accepted, it has to have lots of people participate. So that's why it's so challenging in rare conditions. And I think even if there were enough people in a trial, everyone's different. And those baseline differences and the different trajectories and patterns of change can really affect how it's measured. And you may not get the result you want, even if the drug is actually working, because you're not measuring it in the right way. An example of that is if your outcome is at the end of this, are you able to walk? Well, if you don't get it, but then actually people are saying, but I can move better, I have more energy, it's a negative outcome, but actually it may not be that it is negative. So our approach is to use biomarkers to provide biological evidence rather than clinical evidence of whether there is a treatment benefit. And I think this is being increasingly recognised. And in fact, I think someone mentioned a drug for ALS, tofersen, where in fact it got approval on the basis of a biological measure showing treatment benefit. So I think there's recognition that these can play a really good role in the development of treatments. So we wanted to develop a biomarker panel. So that's a number of different markers all together. So like a suite of measures from one test, to really use across individual child dementia projects to evaluate treatments as they're coming. So for many of the ones that we've heard about today. So just going back to what the definition of a biomarker is, it's just merging two words together, biological and marker. So they're measured in body tissues, so it could be blood, urine, or spinal fluid, depending on where they're collected from. And so they can be used for many different purposes. So you've got to really think about what context you're using them for. They can be used to improve a diagnosis. An example of that might be if someone was identified to have a gene for adrenoleukodystrophy, but they had no symptoms. Could a test tell you when those symptoms are changing or emerging to pull the trigger and start a treatment? Because not everybody that has the gene for ALD is going to get clinical manifestations. So knowing when's the right time, so improving the diagnosis and the certainty about what's going to happen. They can look for shared pathways and targets that are shared for treatment. So I think Tony's talk was a very good example of that. So I don't even need to explain that anymore. Thank you very much, Tony. And then they can evaluate the effect of treatment. So if you've got a drug, if in fact you're targeting the pathways like Tony just talked about, but in fact you measure something in that pathway and it changes, you go, yes, it's actually targeting where we want it to, so let's continue to develop it. Or in fact, it's not doing that. Let's go back and redesign things to get a better drug. So what we're doing is we don't want to do this one disease at a time. We want to take a collective approach and look at what's shared, what's common, but also what could be individual for the childhood dementias. So the first thing we did was to understand the landscape. So we looked at the clinical trials that are currently or have been undertaken to look at which ones actually were using biomarkers, how often they were used, which ones were being used, and what their role was. And so, in fact, there were 262 childhood dementia clinical trials that used biomarkers at some point in the clinical trial, and that was across 49 individual conditions. So most commonly Sanfilippo and metachromic leukodystrophy. But I think importantly is that there's this huge gap, that there's 1/3 of the childhood dementia conditions not actually that we looked for, because we looked when the knowledge base had 70 conditions that had no clinical trial activity that included biomarkers. So it was really important that we weren't just focusing on what we knew, but we were future-proofing for all opportunities. And so, as I previously said, they were looking at different body tissues. They included blood, spinal fluid, and urine. And you can see the graph on the right shows the different conditions that were represented historically in clinical trials and biomarkers. But I think another important point was we wanted to look at the outcomes of those trials and what role they served, but we could only find results for less than half of the studies. Now that could be because they're not yet published, they're still underway, but it's not the whole story. So I think it's really important that if we're doing research, that we are sharing knowledge so that people can learn and in the future. And I think over my time working, we've seen a real shift in this where we really want to be sharing everything that we're learning to help all people in the future. And I think just if people agree with that approach, your voice is important in saying, yes, please, even if it's negative and it didn't show what you wanted to, please share those results so that people don't reproduce that and spend time on that in the future. So that's why I put that point on the slide. So our findings were that there were actually a number of different biological markers that were used, and they sort of ranged from many of the different mechanisms that can happen when you have a child dementia. So aside from the root cause, you can get other things that happen in the brain as it starts to become affected. So you can get inflammation around the brain. So there's markers specific to that were used. There's markers for oxidative stress, for nerves being sick, for changes in the way fats or lipids are handled. And then there's also disturbances in DNA and RNA, which we heard a lot about already today, and also different ways that proteins function in the brain or the impact of them. And when we added them all up together, there were 145 different markers that were used. But I think they were used randomly, not consistently. One study might use one marker, another might use the other. So I think that's why we thought having a panel that was consistently used, and maybe that one marker may not be relevant for one condition, but if you're not looking, you don't know. So we want to develop a panel that can be used consistently in the future. So the key messages, I think, was, yes, we're on the right track. Biomarkers have a role, but I think they're still immature in clinical trials. And to get to the point where they're used in a better way, we need more research to identify them and validate them, find the right ones, and then develop a panel, which is what our research was doing. So the key messages are they're useful, they have different purposes, they do represent the pathways within dementias. There's different pathways, but there's also shared pathways. But we think that there's an ability to address gaps in the development of therapies across all childhood dementias. So I'm going to hand over to Arlene to really talk about where we're up to in this project. Thank you.
Thanks, Michelle. I think this is a really good segue into the second half of our project. Just building on what Michelle said, that in spite of these 145 biological markers being identified in clinical trials, we know that the clinical trials are very homogeneous and highly selected cohorts. So from an experimental approach, we wanted to take this in the lab and really look at biological specimens from our childhood dementia children coming in through the door of the clinic. So this would represent more of the heterogeneity of these conditions and looking into a real world population. Our experimental approach was divided into two phases. So the discovery phase is pretty much going into the dark, fishing in the blind, and looking at all proteins and metabolites which change across these conditions. What are the changes, but what also are the commonalities? And the second phase is to validate our findings by carrying out a second set of experiments. We were only able to do this because of a very diverse team that we are a part of. And this team comprises of neurologists, biochemists, we've got scientists, metabolic physicians, and bioinformaticians. And just to give you a flavor of our research, it really starts at the bedside, goes into the lab, and then feeds back into the clinic. So really from a daily basis, we consent families of children who are affected with these conditions. We collect a biological specimen, as Michelle said, mainly focusing on blood and spinal fluids. We take these to the lab and carry out a suite of experiments. Now, the experiments that we carry out are basically utilizing high throughput advanced technologies. And when I say this, it's the ability to identify a large number of proteins and metabolites from a wide range of samples at a given point in time. So once we've got the data of the proteins and the metabolites, which are altered and which are common, we then put it through a software and carry out some data analysis. And this helps us to identify a set of biomarkers. And really, the whole premise of this approach is to be able to provide a form of personalized therapy to affected children. So really to do this, the first steps were to develop a biorepository. And this was no small undertaking. This was a huge commitment from many clinicians who helped us find these families in so many different clinics and supported recruitment. And really from the get-go, there was a very clear message that we sent out, and that was to be able to unanimously come together, to be able to improve outcomes for affected children, but do this really in a very collaborative manner. We were very fortunate to receive the support, not just from our clinicians, but also from our family members who very willingly participated in the study. And what we really wanted to make sure was that we were not doing unnecessary blood tests or invasive procedures. And so we planned all our specimen collection to be timed with tests for clinical reasons. Having supported blood collection, we know it's no easy task for children and families, and this makes us all the more appreciative of your participation in the study. So far, we have collected 38 biospecimens from the 60 children that have consented to be a part of the study. And as you can see, these biospecimens and all the children are part of the cohort. They fall into different categories of childhood dementia, but also there are specific subgroups that are coming out data from this biorepository. And it doesn't stop there. We continue to track longitudinal collection of biospecimens, especially to look at what are the long-term effects of these treatments and that these children are on. So really, this is a slide to give a snapshot of some of our preliminary analysis that reflects our discovery-based protein study. Now, you'll remember from our review that Michelle specified, around 145 biological measures were identified in clinical trials. But this was in a very ad hoc and disparate manner. And really, because it was such a diverse set of studies that were used, they were not able to come up with a common set of biological markers. So this first analysis really looks at all these conditions under one umbrella. And as you can see on the right, the plot is to give you a flavor of how you can quickly identify changes in this large data set that we were able to establish. So if you can see, the horizontal axis basically represents the magnitude of change, while the vertical axis reflects the significance. The dots on the left are the proteins which are marked in purple, and those were the ones which really went down in expression in our childhood dementia cohort when compared to a fairly controlled neurogenetic cohort. And as you can see on the right, there were 5 proteins which were going up in expression in the childhood dementia cohort as well. So these are the biomarkers that specifically are prime candidates for further investigation in part 2 of our project. And this again is more of an illustration to show that we can identify the common biological measures across this diverse range of childhood dementia conditions. So that was a good proof of concept. We were able to identify similar findings even in our metabolite study. As you can see on the left, the protein metabolites of interest are really separating beautifully into 2 groups, where a set of metabolites are increasing in their expression in the childhood dementia cohort compared to the controls. And again, this goes well in hand with the proteomic findings. So the next steps for us is really how do we take this to a validation phase. We are now developing methods that we can use in the laboratory to validate our findings using a different approach to make sure that what we've identified is not merely due to chance or due to a particular technology that we've used, but is representative of what's happening within these conditions. One important aspect that came out through our literature review and some key conversations with our team was exploring neuroinflammation as an important aspect of childhood dementia. And we are really hoping to pursue this as a sub-project in the coming months. And our work doesn't stop here. We continue to collect these long-term biological specimens, and we want to carry out more experimental work to look at the changes over time. So really the key messages and the impact of our research is generating new knowledge about what biological measures are of pertinence in giving us more information about the progression, treatment effects, and pathological pathways in this wide-ranging spectrum of disorders. And establishing a really comprehensive biomarker panel, not just for diagnostic and monitoring utility, but to really support delivery of a pipeline for new therapies across these conditions through streamlined processes and infrastructure to evaluate therapeutic effect. This work would not be possible without this wide group that we are a part of. We are very fortunate. I would really like to acknowledge our core biomarker team. None of this work would come to light without the Childhood Dementia Initiative. They've been really supportive and helped us right from the get-go of this project to orient our work. We've got a lot of clinicians who are part of the statewide network to thank, as they are so diligent in really identifying the families, engaging with them so that they participate in our study. But lastly, and not the least, our most sincere gratitude is to all the children and their families who've participated and supported our study. And we are more excited in the coming months to share our validation findings with you all. Thank you so much.
Thank you, Arlene. We've run over time already, so we may only have time for one question. And it's handy that it is a biomarker question. So a question from Inika was, have you completed the changes in the biomarkers in the childhood dementia population with other non-childhood dementia neurodegenerative diseases? Are there any unique candidates in the childhood dementia population?
We have a number of candidates. That's a great question. We haven't done it yet, but we'd love to work as a team, and anyone that would like to help us and have input, we're very happy. But thank you for that. And I think as we continue to validate it, we'll definitely do that.
Yep. And there was one other question to Yahoy, and she has already answered that in the Q&A box. So I hope that's reached the person who asked that question. And I just will share my screen again. I would like to thank all the speakers. We're really excited to see the progress and wish you all the best in continuing to push this research forward. Also, thank you to Illumina Corporate Foundation who supported this seminar series. I'm now going to quickly show you a couple of QR codes, so get your phones handy. If you haven't already, please join the Research Alliance so I can keep you up to date with opportunities. And any health professionals that are working with children with dementia, please consider joining our community of practice. And finally, if you can, we would appreciate any feedback you'd like to give us and any ideas for future webinars. That was the last webinar in this series, and we're planning what will happen next. Stay tuned, and thank you again. Have a good rest of the day. Thanks all. Bye.